Somewhere on Earth right now, fewer than 50 people carry something inside their veins that has never been fully replicated, never been synthesized, and can never be manufactured in a lab. It isn't a rare metal. It isn't a lost mineral. It's blood, human blood, but blood so stripped down at the molecular level that doctors who first saw it assumed it couldn't exist. They called it Rh-Null. The world calls it golden blood. And the story of how it was found, why it barely survives, and what it reveals about the hidden architecture of every red blood cell in your body is stranger than most people realize. To understand why Rh-Null broke the rules of medicine, you first have to understand what most people get wrong about Rh blood. When someone says they're Rh negative, they mean their red blood cells lack a single protein called the D antigen, one piece out of a much larger system. Rh-Null is not that. A person with Rh-Null blood lacks every single protein in the entire Rh system, not just the D antigen, but the complete family of Rh antigens present on red blood cells. Depending on how the antigens are counted, that system includes somewhere around 49 to 61 known Rh antigens, and in Rh-Null blood, none of them are there. It is not a variation on a Rh negative. It is the total absence of the entire category. The discovery happened in 1961, in Australia, in the blood of an Aboriginal woman whose sample simply refused to behave. Researchers testing her blood found it didn't react to any Rh antisera at all, meaning it showed no detectable Rh antigens whatsoever. At the time, this wasn't just unusual, it was considered biologically implausible. Doctors before that discovery had assumed a person lacking every Rh antigen simply wouldn't survive to be born. The Rh system wasn't understood as decorative. It was assumed to be structurally necessary, woven into the basic integrity of the red blood cell membrane. And yet here was a living woman walking around with none of it. The medical establishment had to rewrite its assumptions on the spot. The name that stuck came from an Italian geneticist named Ruggero Ceppolini, who coined the term Rh-Null, and the label spread as more cases turned up around the world in the years that followed. A second case identified by physician Philip Levine and his team in 1964, and additional cases surfacing gradually afterward, mostly during routine blood typing, transfusion preparation, or investigations into unexplained blood disorders. Over the following 6 decades, the global tally of confirmed Rh-Null cases has grown with excruciating slowness. Depending on the source and the year of counting, researchers place the number of confirmed cases somewhere between 43 and just under 50 people, ever, in all of recorded medical history. The estimated frequency is roughly 1 in 6 million people, a number so small that most hematologists will go their entire careers without personally encountering a single case. What makes Rh-Null blood valuable, rather than simply strange, is a direct consequence of what's missing. Blood transfusions work because the immune system attacks anything foreign on a donor cell's surface. If a patient carries rare or unusual combinations of Rh antigens and needs blood that won't trigger an immune reaction, ordinary Rh-negative blood may still contain enough residual Rh proteins to cause a dangerous response. Rh-Null blood contains none of them. Because it lacks all Rh antigens, it can safely be transfused to patients with rare Rh phenotypes who cannot tolerate standard blood types. In transfusion medicine, that makes it something close to a universal solvent for the most complicated cases doctors encounter. The patients that ordinary O-negative blood, the standard universal donor type, still can't safely help. But the same emptiness that makes Rh-Null so valuable to others makes it a liability for the people who carry it. The logic runs in reverse once you're the patient instead of the donor. A person with Rh-Null blood can donate to almost anyone, but if that same person ever needs a transfusion themselves, ordinary blood, even Rh-negative blood, will register as foreign to their immune system, because it still carries other Rh antigens their body has never encountered and has no tolerance for. Their only safe option, if they ever require a transfusion, is blood from another Rh-Null donor, and that pool is vanishingly small. Of the roughly 43 to 48 people ever confirmed to carry the trait, only about 9 are known to be active, willing donors, scattered across different countries, most of them essentially unreachable in an emergency. Some of them donate for no one but themselves, banking their own blood in case they ever need it, because waiting for a compatible unit to arrive from another continent is not something a person can do during a car accident or a hemorrhage. The genetics behind all of this trace back to a small cluster of genes on chromosome 1, working alongside a separate gene on chromosome 6. The Rh antigens people are born with come from 2 neighboring genes, RhD and RhC, sitting side by side on the Rh locus at chromosome 1p36p34. But the antigens those genes produce can't simply appear on the cell surface by themselves. They need scaffolding, a chaperone protein, to physically escort them into position on the membrane. That job belongs to a completely different gene, RAG, located on chromosome 6, which encodes the Rh-associated glycoprotein, essential for getting Rh antigens onto the surface of the red blood cell at all. Break either system, and the antigens vanish, not because the genetic instructions for them are gone, but because they have nowhere to go and nothing to carry them. This is why researchers describe 2 separate roads to Rh-Null blood, not one. One is called the regulator type, caused by mutations in the RAG gene itself, and the other is called the amorph type, caused by inactivating mutations directly at the Rh locus that silence the RhD and RhG genes. Because humans carry 2 copies of every chromosome, one from each parent, producing the amorph form of Rh-Null requires an extraordinary genetic coincidence. 4 separate gene copies, 2 RhD and 2 RhCEE, all need to be silenced simultaneously for the phenotype to appear. The regulator form is somewhat more common in the recorded cases, but both routes lead to the same outcome, and both are recessive, meaning a child only develops Rh-Null blood if they inherit a non-functioning copy of the relevant gene from both parents. The inheritance pattern is autosomal recessive, which is why the trait tends to surface more often in families where parents share genetic ancestry. It's a rare enough mutation on its own. For 2 carriers to find each other and both pass it on requires an improbable convergence, which is part of why closely related unions, historically more common in small or isolated populations, appear disproportionately in the family histories of documented Rh-Null cases. None of this would matter as much as it does if the missing proteins were purely cosmetic, a kind of biological blank space with no consequence beyond blood typing. They're not. Rh proteins are structural. They anchor into the red blood cell membrane and link, through a chain of supporting molecules, to the internal skeleton of the cell, interacting with the spectrin-based cytoskeleton through proteins called protein 4.2 and ankyrin, helping maintain the mechanical integrity of the entire cell membrane. Take the Rh complex away, and the membrane doesn't just lose a label, it loses part of its scaffolding. Red blood cells in people with Rh-Null blood come out misshapen, exhibiting stomatocytosis and spherocytosis, abnormal cell shapes that make the membrane more fragile and shorten the cell's working lifespan. The practical result is a mild to moderate chronic hemolytic anemia that many people with Rh-Null blood live with permanently. Fatigue, episodes of jaundice, sometimes an enlarged spleen, sometimes surgery to remove it. It isn't usually life-threatening on its own. It's a slow, ongoing tax the body pays for missing a piece of its own architecture, decade after decade, without ever fully compensating for it. And the Rh complex doesn't travel alone. It arrives bundled with a small entourage of other membrane proteins that seem to depend on its presence to anchor properly, including CD47, glycophorin B, the Duffy antigen, and the LW glycoprotein, all of which are absent or severely reduced on Rh-Null cells. Each of those proteins has its own job elsewhere in immune signaling, cell adhesion, or malaria resistance, and losing all of them simultaneously is part of why Rh-Null blood behaves so differently from ordinary blood under a microscope. It isn't one missing piece. It's an entire missing neighborhood of the cell surface, and the body has to route around all of it at once. For decades, nobody could explain why the Rh proteins existed on red blood cells in the first place, beyond their role in transfusion compatibility and their contribution to the cell's structural skeleton. The deeper answer turned out to be almost unrelated to blood typing altogether, and it came from an unlikely direction: fish gills. Researchers studying how aquatic animals excrete ammonia found that fish gill tissue expressed proteins nearly identical to human Rh proteins, and that those fish proteins were actively transporting ammonia molecules across cell membranes. The Rh protein family turned out to be closely related to the meptope family of ammonia transporters found in bacteria, yeast, and plants, a lineage of molecular gas channels that predates vertebrates entirely. Follow-up work on human red blood cells confirmed the same basic mechanism was happening inside us. Rag, the very protein whose mutation causes the regulator form of Rh-Null blood, was shown to facilitate the movement of ammonia across the red blood cell membrane, functioning as what researchers describe as a gas channel embedded directly in the cell surface. Related work has pointed to a second function layered on top of that one: a coordinated arrangement between Rh proteins and a separate membrane protein called band 3, forming what's been described as a gas exchange metabolon that may assist in transporting carbon dioxide out of the cell. Put simply, the Rh system on your red blood cells appears to be doing double duty. It's part of a genetic identity system used for blood typing and pregnancy compatibility, and at the very same time, it seems to be functioning as ancient molecular plumbing, moving waste gases like ammonia and carbon dioxide across the cell membrane as your blood carries oxygen through your body. That dual role helps explain something researchers noticed when they traced the Rh gene family back through evolutionary time. The genes coding for these proteins weren't drifting randomly, the way junk sequences do. Erythroid AH30 and RHA genes showed evidence of positive selection at specific points across vertebrate evolution, particularly along the lineage leading to humans, consistent with pressure to improve the surface area available for gas diffusion across increasingly specialized biconcave red blood cells. In other words, natural selection appears to have actively refined these genes over millions of years, not because ancient organisms cared about blood transfusions, but because efficient gas exchange was a matter of basic survival, breath by breath, for every animal with blood cells built this way. That's what makes Rh-Null blood such an unusual case study. It isn't simply the absence of a decorative surface marker, the way many blood group variations are. It's the disabling of a piece of cellular machinery with roots stretching back through vertebrate evolution and further still into the microbial world of bacteria and yeast. A person with Rh-Null blood is living, breathing proof that the human body can survive with that machinery switched off. Not comfortably, not without cost, but survive nonetheless, thanks to redundant systems elsewhere in the body compensating for what the missing Rh complex would otherwise be doing. There's one more layer to the paradox, and it's the part that makes Rh-Null blood genuinely dangerous to be born with, in a way that has nothing to do with anemia. Because Rh-Null red blood cells display none of the Rh antigens, a person's immune system has never learned to recognize any of them as self. If that person is ever transfused with ordinary blood, even blood that's Rh negative in the conventional sense, their immune system can respond to it as a hostile invasion, producing an antibody researchers call anti-Rh29, an antibody directed against essentially the entire Rh complex at once. That immune reaction is why only Rh-Null blood remains compatible for any future transfusions once a person has been exposed. It's a closed loop. The rarer the blood becomes worldwide, the more precarious it becomes for the very people who carry it, because their survival in a medical emergency depends entirely on a donor pool smaller than the population of a single classroom. That fragility is why blood banks that manage to identify an Rh-Null donor treat the discovery the way a museum might treat an irreplaceable manuscript. Managing Rh-Null blood on any practical level requires international cooperation, cryopreservation of frozen units, careful autologous donation planning where possible, and ongoing screening of relatives who might unknowingly share the trait. Units of Rh-Null blood have, in documented cases, been flown across oceans to reach a single patient in crisis, arriving just in time and sometimes not soon enough. There is no synthetic substitute. There is no way to manufacture it in bulk. Every unit that exists came from one of a small handful of human bodies, willingly given, carefully frozen, and tracked with a level of precision usually reserved for organ transplants rather than blood donations. Geographically, Rh-Null cases resist the kind of tidy narrative that other rare traits sometimes offer. Cases have been documented on every inhabited continent and across a wide range of ethnic backgrounds, with a slightly elevated number of reports coming out of parts of South America, though nothing resembling a strong or consistent ethnic pattern. It shows up in isolated pockets in Japan, in Europe, in the Middle East, in Southeast Asia, and in indigenous Australian communities, tracing not a single point of origin, but dozens of independent genetic accidents, occurring quietly and separately across human history whenever two carriers of a silent mutation happen to have children together. There is no ancient tribe of golden-blooded ancestors scattered across the globe. There's just an extraordinarily rare, recessive mutation capable of arising wherever the genetic dice happen to land the same way twice. The science around blood group discovery hasn't slowed down either. In June 2024, researchers announced an entirely new blood group, temporarily nicknamed Guada-negative, identified in a single woman from Guadeloupe, presented at the International Society of Blood Transfusion Congress in Milan. It's a reminder that even after more than a century of blood typing, after the ABO system, after the discovery of the Rh factor in 1940, after Rh-Null itself in 1961, the human genome is still capable of producing configurations nobody has ever documented before. Each new case adds another data point to a map of human blood variation that is nowhere near complete. What Rh-Null blood ultimately demonstrates is something larger than a curiosity for a trivia night. It shows how much unnoticed machinery is doing quiet, essential work in a system most people never think about beyond glancing at a blood type on a donor card. It shows that a mutation doesn't have to be common to be scientifically important. Sometimes the rarest possible version of something teaches researchers more about the ordinary version than millions of typical cases ever could. And it shows the strange, double-edged nature of biological scarcity. The same absence that makes a person's blood almost universally useful to others is the very thing that leaves them with almost nowhere to turn if their own body ever needs help. Fewer than 50 people on Earth currently live inside that contradiction, carrying something between a gift and a vulnerability in every vein, a version of human blood so rare that most of medicine will only ever read about it and never actually see it.